Engamana, engareo, ro rakatidama, tena koto, tena koto, tena koto. Norera, ko aio, ko Harleen Hain, takawingawa, ko te tumuakio te whariwana ka oaho. Namihi nui kia koto, norera, tena koto, tena koto, tena tato katoa. Good evening, everyone. My name is Harleen Hain, and for the next few days, I am the Vice Chancellor here at the University of Otago. And it gives me great pleasure that my final introduction for an IPL tonight is in honor of Professor Fiona McDonald, someone who I have known for a very long time. On behalf of the university, I would like to begin my comments this evening by welcoming some special people in Fiona's life who are here with us this evening. Her husband, Graham. Yep, here. Their children, Matthew, Daniel, and Laura. Her parents, Gordon and Elaine, and her sister, Claire, and Claire's partner, Simon, and children, George, Allie, and Lily, and her friends, Helen, Lori, and Fran, and her parents-in-law, Ray and Marilyn, are joining us on the live stream. Now, it's absolutely fantastic that all of you could be here this evening. The presence of so many special people in Fiona's life is a very clear signal of the importance of tonight's event. As I look around, I'd also like to acknowledge professional and academic staff and students from the University of Otago and members of the wider Dunedin community, as well as our Chancellor, Dr. Royden Somerville, who have all come to share in this celebration. On behalf of the university, no mai haramai, welcome. Now, these IPLs are a time of great celebration for us here at the University of Otago, and I'm delighted to see so many people who are here to mark this incredible landmark in Fiona's amazing career. Now, as you will have heard me say many times over the last decade, getting here is never easy. A candidate for a professorship must demonstrate excellence across all aspects of their portfolio: teaching, research, and service. They must not only meet Otago's exacting standards, but their work is also scrutinized by internationally recognized experts in their area of research. Now, as you will learn tonight, Professor McDonald has an international reputation as a first-class researcher. Her research has all the hallmarks of a well-respected scientist. She's published in top international journals in her field. Her work is highly cited, and over the years, she has earned significant external funding in a highly competitive research environment. She has also made a substantial contribution to the training of research students. As you will also learn this evening, Professor McDonald is a passionate and gifted teacher. She excels not only in traditional lecturing in a classroom, but she has also been heavily involved in curriculum development. She's played a significant role in the revision of our very, very large first-year health science program. And as the chair of that committee, I have found it an absolute pleasure to work with her. Her care and her compassion for our students is palpable, and her calm, university-centered approach to decision-making has been outstanding. Now, throughout her career, Professor McDonald's service to her discipline, to her department, and to the university has been numerous, substantial, and valuable. But last but not least, Fiona is an extremely collegial member of our academic community. She is never afraid to speak her mind, but she always does so in a careful and considered way. When Fiona has something to say, it pays to listen. We're extremely lucky to call her one of our own. So Fiona, on behalf of the University of Otago, it gives me great pleasure. I'm going to try desperately not to make you cry by crying. To congratulate you on your very, very, very well-earned professorship. I feel personally proud that you were one of the very first senior lecturers that I mentored when I was the head of the psychology department. I really look forward to your lecture this evening, and I'm now going to call on the slightly less emotional Professor Brian Hyland to tell us just a little bit more about Fiona's path to professor. Norera, tena koto, tena tato katoa.
Thank you, Professor Hain. Tēnā koutou, tēnā koutou, tēnā koutou katoa. Welcome, greetings, greetings, greetings to you all. As Professor Hain just said, my role is to introduce Fiona's inaugural professorial lecture, and I want to spend a few minutes reiterating and expanding on some of the points raised already. So it is my great pleasure and privilege to introduce Fiona, and also to add my congratulations to this well-deserved promotion. So Fiona is a graduate of this university. She graduated with a BSc, first-class BSc honours in biochemistry, during which she won the Edson Prize in 1989, and she went from there directly to a DPhil training at Oxford, which she completed in 1993, having won a Commonwealth PhD scholarship from here and a St Hugh's College, Mary Lunt Scholarship from Oxford itself. She completed her PhD in perfect timing, 3 years, 1996, and went from there to the Howard Hughes Medical Institute for a postdoctoral fellowship in Iowa, United States. From that, she was immediately recruited to a lectureship at Victoria, University of Wellington, where she excelled and was promoted to senior lecturer in 2001. That same year, she was headhunted here to Otago, to the Department of Physiology, the year 2001 when she was promoted to senior lecturer, she was headhunted here, and again, performed exceedingly well, was promoted to associate professor in 2009. Now with promotion to professor, we you know, as has been said already, we know that reflects excellence in performance, but also the step up to leadership that's sustained over an extended career beside continued excellence in performance. So it's a major achievement. So just looking at that a little bit in terms of service leadership at the university, Fiona has served as deputy head of department, looking after teaching side of things from 2013 to 2017. And I know, personal experience, that that deputy role in a department the size of physiology's is the equivalent of a headship, a huge responsibility which she undertook with calmness and effectiveness, and led the department in that aspect exceedingly well over that time. She's moved from there to the headship, full headship of the department since 2018. In teaching side of leadership, at the same time as she was head of department, she was the chair of the papers committee for the Hubs 191 and 192 papers over the period of the recent major revisions to those papers. Considerable input and impact on teaching to the single largest classes, the largest classes here at Otago. Very important course in the context of the Otago teaching and learning experience. She's been the chief course advisor in the Department of Physiology over the same time period, since even longer, since 2014. And prior to that, back in history, she was the course convener for Biology 115, which those of you who have been around long enough will remember was the predecessor for Hubs, involved a very large course looking after managing staff, effectively a department in many respects. So a long-standing tradition of excellence in leadership in teaching at the university. And many more, you know, I could go on and on with the roles and responsibilities that Fiona's taken on and always done exceptionally well over the years. Been mentioned, teaching performance, Fiona, you know, outstanding teacher in the department. She was a recipient of the Biomedical Sciences Distinguished Academic Teacher Award in 2017, reflecting that. And at the interface of teaching and research, as again mentioned, she's mentored more than 40 postgraduate students in her time as an academic, including 5 masters and 14 PhD students, and all of them complete an excellent time and with wonderful outcomes for those students. In terms of research, outputs and recognition, 2005, she won the New Zealand Association of Scientists Research Medal. 2011 was awarded a Fulbright Scholarship. And she's funded her work with a succession of grants and engaged in highly fruitful national and international collaborations. As a result of all that, she's published as senior or first author on multiple peer-reviewed papers in high-impact scientific journals, including Cell, Journal of Clinical Investigation, Proceedings of the National Academy of Sciences, American Journal of Physiology, Journal of Biological Chemistry, a wide range of journals reflecting the breadth of her research interests and outputs. Also been invited or submitted and had accepted for publication many major reviews in her field, including in the American Journal of Physiology. There are multiple sections in that journal, so she's included reviews in circulation physiology, in cell physiology and renal physiology, and also in the journal Heart Failure Reviews. And of course, review articles very widely cited and important benchmarks in the field. Interestingly, and it's already been mentioned, active in teaching and learning related research and is published also in the educational literature. So the research directions, if I can summarise for you, I don't want to take up too much of her talk, but Fiona's research focused on the control of blood pressure by the kidney, and salt, or sodium in other words, in the blood can have an important impact on blood pressure. Fiona's work addresses this at the cellular and molecular levels. In particular, she'll be talking, I think, quite a lot probably today about a particular component of cell membranes in the kidney, the epithelial sodium channel, which is a critical mediator of sodium effects in the kidney. And her work, I'm sure I'm going to leave her to describe how her work relates to the function of this channel and how important it is for regulating normal blood pressure. She's also contributed in that line to work uncovering how mutations in that channel contribute to a specific clinical condition where very high blood pressure occurs, called Liddle syndrome, and reflecting that channel proteins almost never have a single function in the body. More recently, her work has expanded into how changes in that sodium channel activity can affect breast cancer cell proliferation and migration. And these are important potential targets, I think, for anti-cancer therapy into the future. So in her talk this evening, Fiona, I think, will share with us aspects of her career journey, key research results, as well as perhaps, I hope, delving into reflections on teaching and learning. Once again, it's my pleasure and privilege to introduce Professor McDonald to you to deliver her inaugural professorial lecture. Tēnā koe, Fiona.
Tēnā koutou, ko kapawai tō mana, ko mataawa te awa, ko Pakeha toko iwi, no Roxburgh aho, ko Gordon, rawa ko Elaine, o ka matua, ko Fiona McDonald toko ingoa, no rai rā, tēnā koutou, tēnā koutou katoa. Thank you, Professor Hain and Professor Hyland, for your very kind and generous introductions. And I'm really privileged to be able to share with you my journey to being able to deliver this IPL. Along the way, I'll mention just a few of the people who have been important in my journey. Friends, family, students, and research staff, collaborators, colleagues, and mentors, they're all part of my success. And I guess I'll start by saying that my story begins in Roxburgh. You've probably passed through it, going to Alexandra or Wanaka or Queenstown. It's the home of Jimmy's Pies. I worked there for several summers. I grew up on the east side of the river. So this is a picture from the 1970s, and my parents ran a sheep farm here and an orchard. And it was a lovely way to grow up. Grew up there. with some, her farm was in the family for a few generations. So, hence the architecture. I grew up with my parents and two sisters, Claire and Lauren, and we travelled down this road to the end to catch the school bus, and we attended Roxby Area School. Here's a photo from me in Form 1, I believe, with braces. This area is not part of our family anymore. If you go past and go up Kynaston Road, this is now large-scale orchards, mainly apples and cherries. So I left Roxburgh actually pretty young. I was about 12 and a half, and I moved to Ōtepoti, Dunedin, where I came to high school. I was a boarder at Tolkan and went to St Hilda's. This, as you can imagine, was a pretty formative experience. Leaving home, having to become independent, making decisions for myself. But on reflection, there's a couple of other things that I really felt set me up quite well, and that was gaining excellent study skills. So we had supervised compulsory prep for 2 hours a night on weekdays, and that carried on right through the 5 years of high school. And so I developed really excellent study skills, which put me in good stead for later study. I also realised how much knowledge there was out there and loved learning about so many diverse topics. You might be surprised to know that I failed my first science test in Form 3, and I was probably more interested in history and English for the first few years of high school. But I took biology and chemistry right through to Form 7, and I had a wonderful chemistry teacher, Mrs. Gucci's, and I did fall in love with chemistry at that time. I did consider going to Teachers College at the end of high school to become a primary school teacher, but in the end I decided that university was the place for me, even though I was first in my family to attend university, so I didn't really know what to expect. However, I enrolled in a BSc majoring in chemistry. So along with my chemistry papers, I was taking biology and maths and computer science papers, and as part of one of those biology papers there was a biochemistry component. It didn't have its own subject at first year like it does now, and that really hooked me because it was chemical reactions that were related to how cells worked and how our bodies worked. So I ended up completing a major in both chemistry and biochemistry, and when it came to my 4th year I had to decide which department to do my honours degree in. Well, I'd really found that I loved thinking and working with proteins. So our proteins are kind of the structural components that our genes code for, very important for how our cells function, and I was interested in how those proteins sort of moved around the cell, that was sent to the right location, and how they were regulated. So for my honours project, I was matched with Murray Grigger, shown here, and he had a lab at that time shared with Warren Tate, shown here, and the adjoining lab was Clive Trotman's. And there was a wonderful, really supportive group of students and staff in that lab area, and it was a great introduction to research. Murray had an interest in how during lactation, the iron levels of the milk remained high enough to feed the pups, and my project was looking at a protein called ferritin, which was is a storage protein for iron. It turned out that it probably wasn't the most important factor for maintaining iron levels, but certainly my work contributed to Murray's ongoing work, and it looked like the protein transferrin, which circulates in the blood carrying iron, it seemed to be really important for the milk to have enough iron for the pups. So I graduated in 1989, and here's a picture of me in the biochem department just before heading down to the town hall. Now, between my 3rd and 4th years of study, I had a wonderful opportunity to go to Canberra over the summer and do a summer research project at ANU. I worked in Dr. Mackenzie's lab, purifying proteins from hen eggs for ongoing analysis. Now, I only had this opportunity because my chemistry friend Jenny would go walk through the archway, and she saw a notice there that said apply for these summer research scholarships. She was kind enough to share that with me, and we both applied and were both accepted to go to Canberra. And we joined this amazing group of summer scholars from Australia and New Zealand. There were 60 or 70 of us. We were all living in the same college and then doing research projects around the university, and that's me in the circle. And I think that was a really pivotal moment because the conversations between all these different students, including my good friend Eve, who I met there, they were all discussing doing their PhDs in the UK or the US, and I sort of hadn't thought about what I'd do after my degree. And that really put in my mind that, I could actually do an OE by going overseas to do a PhD. So during my honours year, my supervisor, Murray Grigger, was really encouraging, and I applied for several scholarships. And as Dr. Hyland mentioned, I was offered a Commonwealth Scholarship in the UK at the University of Oxford, which I took up in Michaelmas term in 1989. I joined the lab of Dr. John Heath, shown here, and he was a relatively new lecturer at that time, but he had gathered around him an amazing group of developmental biologists, people who understood how to make mouse transgenic models, and people working on signalling pathways. Again, it was a wonderful environment, very supportive environment to do my PhD. John asked me to study a protein called fibroblast growth factor 4. Very, very little was known about it, but it did seem to be important during development. And there was a new technology that was, really very new, and that was making mice that had a single gene deleted. Quite a long process, but he had all the tools and we had the support, so I started trying to make this knockout mouse model so that we could find out what FGF4 did during development. Well, I was worked very hard. I made multiple targeting constructs and was able to delete the gene in cells and culture, but unfortunately we didn't get to the mouse to make the mouse model. However, I did lots of other work on FGFs and FGF receptors and was able to submit my thesis in the middle of 1992. And I, like many students, perseverance is a skill that you learn when you're doing your PhD. I did a lot of screening, a lot of growing cells, and a lot of negative results. And I think that does set us up well. We have the negative results, but we also have those shining moments where we get the result that we were looking for. I was also, of course, able to experience the unique environment that is Oxford, living in a college that was St Hugh's, rowing for my college, and another real, looking back, key event was the lifelong friends that I made there. So this includes Julie, Helen, Catherine. This is not from the 1980s. This is where we actually had a reunion a few years ago, the first time we'd been together since we were studying in Oxford. And it was Helen who was the one who taught me how to make all these transgenic mouse models. And as a little bit of a payback, I was able to provide some reagents to her, and it was her work who showed that FGF4 was really important for limb development during embryonic development. So I finished my thesis, I graduated, and again, end of a degree, had to think about what to do next. Well, I decided that I wasn't quite ready to come home, and that while I loved developmental biology, I wanted to do something that was a bit more, had a personal connection. So we have cystic fibrosis running in our family, and my little sister died with CF when she was 11. So I thought this might be a great time to look to work in a cystic fibrosis lab. And fortunately, I'd been asked to present my work at a Cold Spring Harbor conference in the US, so I travelled there and visited a few labs, including the lab of Mike Welsh at the University of Iowa. And this, he is a giant in the cystic fibrosis field. He showed that the protein that is mutated in cystic fibrosis is a chloride channel, and that actually the major mutation causes the protein to be mistrafficked. It's not taken to its correct location in the cell. So it was a really, I was really looking forward to working with Mike, and fortunately he did offer me a position. However, I ended up not working exactly in the CF field, but this is where I started working on the epithelial sodium channel, or ENaC. So I moved to Iowa in March 1993, so I went from balmy central Otago to the Midwest. Here's Iowa, well known for its cornfields, to, I don't know, a couple of feet of snow. And Mike, he picked me up from the airport. That was pretty cool. He took me out for breakfast, and he put a paper in front of me on the table. And this paper had been published in Nature about 3 weeks beforehand, and it described the cloning of an epithelial sodium channel. And it was a channel that was known to be present particularly in the kidney, and no one had been able to clone the gene for this protein. And it was actually a Herculean task by Cecilia Canessa and Bernard Rossier's lab. And this protein was predicted to be really important for controlling body fluids and to have relevance for cystic fibrosis and also for blood pressure regulation. So Mike asked me to clone the human equivalent of this gene because Canessa's gene was from rats, and so that was my first task. But before I describe that work, I'm going to give you a short lesson in kidney physiology. All right, so you've all got two kidneys, and they have a very important role in filtering the blood. They take your waste products, and those waste products move to the bladder so they can be expelled in the urine, whereas other components are recycled back into the blood. Now, in each of your kidneys, you have about 1 million nephrons, and these are small tubes, and I have just a demonstration of what a nephron might look like. So it's a tube, this clear thing here, and it's closely connected with blood vessels. And there's a filter, but like a sieve or a colander at the top here, where small chemicals can move from the blood into this tube. And it points along the way. Some of those chemicals, like sodium or water or glucose, can be brought back into the blood, and what comes out at the end is the urine. So if you opened up this tube and put it flat, it would look something like this. So around the tube are epithelial cells, and then this is the components of the blood moving down the tube and eventually forming the urine. Well, small chemicals like sodium and water and glucose, they can actually be rescued. So here's a sodium molecule moving through that epithelial cell and back into the blood. But some of the sodium will inevitably be lost in the urine. Water can do the same thing, and it can be recycled into the blood. So that means that the body can collect back what it can't afford to lose. So sodium, water, glucose, and a lot of other ions and chemicals. But waste products can be expelled in the urine. So how does this relate to blood pressure? Well, here. If you increase the amount of sodium that's moving into the blood, then water will be pulled along by osmosis, and this will increase the volume of blood in your blood vessels, and that will increase your blood pressure because your heart has to work harder to pump all of that blood around your body. Now, high blood pressure is a risk factor for heart attacks, kidney disease, stroke, and other cardiovascular complications. Normally, our body keeps our blood pressure at a normal range, but your kidney has an important role in controlling blood pressure through this sodium reabsorption pathway. Of course, your heart, blood vessels, and brain and other organs are also important. So these epithelial cells, these are facing the external environment. So they only pick certain chemicals that can be brought back into the body. They have to be very careful about what comes back in. So there's a gate here that allows only the sodium to come back into the cell. And that gate is this epithelial sodium channel, which allows sodium to move into a cell, and then that sodium can move out into the blood via a different mechanism. So the epithelial sodium channel, ENaC, we now know, is actually made up of 3 subunits, alpha, beta, and gamma, and together they transport sodium into a cell. And the prediction was, when this channel was originally cloned, that if there was too much ENaC sitting in those epithelial cells, you get too much sodium moving into the body, and that might cause high blood pressure, or if there was too little, then all the sodium was being lost in the urine, and that would probably cause low blood pressure. However, in 1993, we didn't know a lot of these mechanisms. And remember, I said that my first task as a postdoc, I had to clone the human gene for this epithelial sodium channel. Now, the human genome wasn't published, so I had to use traditional cloning methods. Fortunately, the rat DNA sequence for a gene is normally relatively similar, normally about 80% similar to human gene sequences. So I used part of that rat ENaC gene, and I screened a human kidney DNA library, and I pulled out the gene for human alpha ENaC. We had to test whether it really was the human version of the gene. We found it was expressed where we would expect it to be. We found it in kidney, colon, and lung. And then, working with my good friend and colleague Pete Snyder, he carried out some sodium transport measurements, and we used a very well-known system of using eggs from Xenopus frogs, and we could also use a specific drug, amiloride, that was known to inhibit this channel in vivo in the kidney. So this is a current trace over time, and you can see that this line represents sodium moving through a channel, and when amiloride is added, the current is decreased down to baseline levels, and that's reversible, and that gave us great confidence that we cloned the human version of this epithelial sodium channel, and we're able to publish that. Well, as I said, there's actually 2 more subunits, and Canessa et al. Had published in 1994 that there were 2 more subunits, so we went, right, we better clone the human versions, and my colleague and friend Maggie Price did one, and I did the other, and we were able to show that we could put all those subunits together and give a sodium channel current that looked pretty much like what had been measured in vivo. So a large amiloride-sensitive current was generated. This was, it was a very exciting time. There were a lot of people jumping on the ENaC bandwagon. Things were happening quickly. People were looking at how the channel was controlled and whether it was implicated in high or low blood pressure conditions in humans. And that came pretty quick. The first paper to show that ENaC was, this is really a pivotal paper because it showed that mutations in ENaC could cause Liddle syndrome, which is an inherited form of high blood pressure. And the mutation was specifically found in the beta subunit. It has a tail, and when the Liddle's mutation was present, that chopped off the last 75 amino acids. And that paper was published from Rick Lifton's lab in 1994, but there was no mechanism. So we quickly made some mutants, and because we predicted that there would be increased sodium transport through ENaC, and that's exactly what we found. So this is, again, it's a bit blurry, but this is current measurements from the wild-type ENaC channel set at 1. And when we made the exact Liddle's mutation in the beta subunit and put it together with the other normal subunits, we saw a massive increase in current, nearly 2-fold. And that can explain, or go partway to explain, why this mutation in ENaC caused the high blood pressure. But I will come back to that. On the opposite side of things, if Mike Welsh was very keen to make some mouse models because he wanted to know what would happen when we deleted the ENaC genes, and I had tried to make another knockout mouse. Fortunately, the technology had moved on quite a lot and we were successful making that model. And it actually recapitulated the symptoms of a human disorder known as pseudohypoaldosteronism, or PHA, which is an inherited form of low blood pressure, where your sodium levels are low in the blood and you're wasting all that sodium into the urine. And that had quite severe consequences in the mouse model. Okay. So as I said, we were doing a lot of different work in Mike's lab. It was great to be there. And I had thought I'd carry on a little longer, but my mum stepped in. She sent me a clipping from the ODT, lecturer in cell and developmental biology wanted at Victoria University in Wellington. I sent off my CV, didn't really think much more about it, but then one day in the lab there was a phone call, and it was John Miller from Vic, and he'd called up to interview me. No preparation. I don't remember many of the questions except one where he said, "Aren't all those molecular techniques really expensive?" And I said, well, if I was appointed, I'd just apply for external grant funding to pay for it. Very naive. And that would cover the costs. Well, hung up. Didn't think much more about it. But then a couple of days later, John called up again and offered me the lecturer position. It's quite different to the interview processes we run today, isn't it? Well, and I had to decide, I'd had nearly 3 years as a postdoc. I could have gone on and learnt a lot more in Mike's lab. But I also weighed up that finding a lecturer position in New Zealand in my field was going to be hard. And I also thought back to my commitment when I'd taken up the Commonwealth Scholarship that I was committing to come back to New Zealand at some point. And so I said yes. And in 1996, I moved back to Wellington and joined the School of Biological Sciences. Now, there's a couple of reflections from that time. Remember, it's a new lecturer position. And the first is around curriculum development. Well, it turned out that the cell and developmental biology focus was new for the school. And it also turned out that I had to develop a whole new third-year paper to start in July. I arrived in March. 24 lectures, 6 labs. But it got it done. It was great with John's help and also one of my other mentors there, Bill Jordan, pulled that off. Students really enjoyed it. Not then next, John and I had to develop a whole new second-year cell and developmental biology paper that rolled out the next semester. Another 6 labs and 24 lectures for each of us. However, perhaps that's what set me off on the curriculum development, but it was a lot of fun, and again, we got good evaluations of that course. Okay, but I also, of course, had to launch my independent research program, and I decided to focus on identifying proteins that bind to ENaC, thinking that they would be regulators of ENaC and potentially blood pressure. Again, mentored by John and Bill Jordan, I was fortunate to be awarded an HRC project grant and had other local funding. Also fortunate to hire a research assistant, Tracy Farr, and started a productive collaboration with Sean Lott, who was at Massey University at that time. And with Tracy's help, we worked on an unknown protein that I'd found in Iowa to interact with ENaC. It turned out to be a protein called NED4, and actually others in the field had also found that this NED4 protein bound to ENaC. So we showed that this NED4 protein bound to each of the subunits, and also, very importantly, when we studied sodium transport with current measurements, we could measure plenty of current when we just expressed ENaC alone, but when we added in the NED4, suddenly current decreased. So that told us that NED4 was somehow inhibiting sodium transport, and one of our goals was to find out the mechanism for that. However, there was a Physiological Society meeting at Victoria, and I met Tony McKnight, who is a past HOD of physiology, and he invited me to interview for a position here in physiology at Otago. I think my friend Kath Dickinson has something to do with that as well. Anyway, I interviewed and an offer was made, and it was a really appealing move, because while I was pretty competent in doing a lot of biochemical and molecular techniques, I did need some bit more help with the function, and there were several great epithelial physiologists in the department, not only Tony, but also Grant Butt, Kirk Hamilton, and John Leader, and so this was a good move for me. So I moved down to Dunedin. Actually, it's 20 years ago, I think first week of April. And I continued looking at control pathways for ENaC, and I'll just briefly mention a couple of those results. First of all, I was able to recruit an excellent PhD student, Dominic Weemuth, and he carried on looking at this NED4 protein that we'd found interacting with ENaC. And as I said, others in the field were publishing in this area as well, and it turned out that NED4 is an enzyme, and it can catalyse the attachment of a protein called ubiquitin onto ENaC. And Dominic had the job, or we came up with this project where we would just look at the membrane population of ENaC, and he showed that NED4 could bind to that surface population of ENaC, tag the subunits with ubiquitin, and that would cause their internalisation into the cell, so they couldn't transport the sodium anymore. And from there, the channel could be sent to cellular compartments and eventually degraded. And so that meant that you could lower the amount of sodium coming into the cells, and this, in the kidney, this would lower blood pressure. This also has relevance going back to the Little syndrome story. I told you how in Little syndrome, the mutation removed the last few amino acids from the beta subunit. And it turns out that this is the exact area that the NED4 protein binds to. So we think that the lack of NED4 binding has a really important, it's helped us understand the phenotype of Little syndrome, and I'll show that complicated cell model again, but essentially this NED4 protein cannot bind to ENaC when there's a Little's mutation, so that means that the channel can't be internalised from the membrane, and it can't, oh, I've got some crosses, it can't be sent for destruction, so that all those channels stay at the cell surface, and they're all moving sodium into the cell, and all that sodium moves into the blood, and that increases blood pressure, which is causing Little syndrome. In a second parallel set of projects, I again was fortunate to recruit postdoc Wolfgang Biasio, and he was a fabulous molecular biologist. And he set about doing something called a use-to hybrid screen, which is essentially where you really don't have a hypothesis of what is interacting with ENaC. It's a wide open screen. And we actually chose to use a fourth human ENaC subunit, the delta subunit, that can substitute for alpha, because no one had really looked at this protein. And Wolfgang pulled out a protein which at that time was known as MUR1, and there was a single published paper on this protein, and it was about Bedlington Terrier dogs. When they had a mutation in this MUR1 protein, they started to store copper in their liver, and that caused copper toxicosis and death. So clearly this protein was pretty important, but it didn't really relate to our sodium channel. But we did show that MUR1 could bind to the channel and also change the level of sodium being transported through the channel. To complicate matters, it turned out that MUR1 had brothers and sisters. It was a protein family, and that was described by Ezra Burstein, that there were 9 other members. They had to be called something different, so the name was changed to the COMD proteins. Now in 2011, I started to think it might be quite good to take some research and study leave. So I actually approached Ezra Burstein and asked if I could come and work in his lab so that we could learn more about the COMDs. because I'd had various students working on the different COMDs. They all seemed to interact with ENAC. They all altered sodium transport through the channel, but we still didn't really know what they did. So Ezra said yes. And in 2012, our family moved to Texas, where I worked with Ezra at UT Southwestern Medical Center. So went down, very different climate in Dallas, and really enjoyed our time in the Lone Star State. This was supported by a Fulbright Senior Scholar Award and also the University of Otago. I realised there was a theme developing when I put this talk together. I was working on another knockout mouse, this time on a COMD protein, and Ezra had just developed this model and it needed someone to characterise it. So that was me. Again, there was a fabulous group of scientists to work with, lots of support, and I did work at the bench pretty much for the whole year. And I found that the COMD knockout was an embryonic lethal, and so that meant that COMD10 was so important that embryos couldn't get through to full term. And I think, I worked out that it was needed around late gastrulation. That was the pivotal time. But I also found using cell culture experiments that the COMD10 was also needed for proteins to move to the correct place in their cell, which was a clue for actually what the mechanism we now know that the COMDs work through. So that was 2012. When I returned in 2013, I was really fortunate to hire fabulous ARF, Tanya Chung, and also recruit a PhD student, also fabulous, Adam Ware. And we set out to build on the work that I'd been doing in Texas, and with the collaboration with Ezra. And we now know that these COMD proteins, their role seems to be to rescue ENaC from destruction. So I'm going to show you another cellular model because I can actually fill in another part of this pathway. So I showed you that NED4 was a negative regulator of ENaC, and it could drive ENaC coming in from the membrane into the cell and be degraded. Well, this pathway can be bypassed because these little ubiquitin tags can be clipped off, and that leaves ENaC available to be rescued. And it does that we think, by binding these COMD proteins, and they in turn bind to a recycling complex such as retromer, and that means that this whole sodium channel can move back to the cell surface and do its job again, bring the sodium in, and help to be regulating blood pressure. So a lot of our work has supported this concept that this surface population or membrane population of ENaC has to be exquisitely controlled, or else if you have too much, you get high blood pressure, too little, you get low blood pressure. Under normal conditions, we think that this NED4 protein keeps relatively low levels of ENaC at the membrane, but that can be inhibited if more channels are needed, or this recycling pathway can also be increased, so you've got the right number of ENaCs. So I'll give you a little break from ENaC and just mention a little bit about our research into teaching and learning. So as teachers, we all want to give our students a great experience, but we also want them to gain a deep understanding of the field that they're studying so that they can go on to develop new knowledge, solve problems, and develop their own critical analysis skills. So I had the opportunity when we were developing the human body systems papers back in 2006, 2007, to study the effect of curriculum changes and the time of study on student approaches to learning. Now, you might not have thought about this before, but maybe think about for yourself how you go about learning. Do you take a surface approach, where you just memorize everything, don't really think about what it means, and put that down on paper? Or do you take a deep approach to learning, which we would prefer, where you're seeking meaning in that information and relating ideas and getting excited about what it means for the wider picture? There's also, in the literature, defined a strategic approach where students will change their approach to learning depending on external pressures, like, is this assignment due, or do I need to do these other things? So when the Hubs papers were developed, I ended up being the convener of Hubs 192, and John Reynolds was the convener of Hubs 191. And we were really fortunate to collaborate with Rachel Spronkin-Smith and Carol Bond and others from HEDC, and we looked to see whether that new curriculum we were developing could promote a deep approach to learning in our students. And so we followed this cohort of students not only through 2007 but also into 2009, and we had those students fill out a reputable survey which gave us some clues about their approaches to learning. And even within a single year, we saw that the extent of deep learning increased, as did the strategic learning, and their surface learning approaches seemed to be less. And I guess gratifying, at the end of their studies, these third-year students were using deep approaches to learning to a great extent, as well as their strategic learning, but they were still doing surface learning as well, where they had to memorise some material. So that resulted in a couple of publications, and that was a nice parallel pathway into researching into those learning approaches. But I'm going to finish up now with our newest research area, which, as Professor Hyland indicated, is looking at ENaC and breast cancer. So you've got mammary epithelia in breasts, and ENaC, not surprisingly, is expressed there. Now, it was on the advice of a visiting scientist from UQ, Greg Monteith, who works on ion channels in cancer, and he suggested that we should look at ENaC. And he pointed us to a database of women, and it's an international database, which collects information about women with breast cancer, and it looks at which genes are expressed and the level of that gene expression. And just from interrogating that database, we could see that those women with breast cancer who had a poor prognosis had very low ENaC levels, and in the opposite scenario, high levels of ENaC seemed to correlate with good prognosis. So we have a team now in physiology, some shown here, and fantastic collaborators in pathology. And Adam Ware spearheaded this project, and we cobbled together money from various different funding agencies and started looking to see whether ENaC really could have a role in breast cancer, and we used cell culture models. So I've talked to you about ENaC. It's an epithelial sodium channel. It's found in epithelial cells, and epithelial cells have a very distinct not only function but shape and structure. They are very tightly bound to each other. They're facing the external environment, so they have to control what's coming into the body, and they sit on a dense fibrous mat that they're joined tightly to. They're also polarized, which means that proteins like ENaC are only found on one membrane or the other. ENaC always faces the external environment, but other proteins, different proteins, are down here. Now, in cancer, epithelial cells can change their morphology to a state that's referred to as a mesenchymal cell. So they can lose their connections with their neighbours and with that underlying matrix, and they can also take on different properties such as a high rate of cell division, which is a hallmark of cancer. Whereas epithelial cells tend to have a pretty low rate of cell division. So we asked the question, well, what would happen if we changed the levels of ENaC? Sorry, one more arrow. It turns out that this process is reversible, and it's referred to as epithelial mesenchymal plasticity or transition. So it is possible for cells to go one way, and but also to move back from a mesenchymal to a more epithelial state. So we set up the experiment where we increased the numbers of ENaC channels in the mesenchymal breast cancer cells. So we added more ENaC, And that seemed to push them back into this more epithelial state, probably more correctly to say that they became less mesenchymal, and they had a lower rate of cell division. And I'll just show you one result from this, where Adam carried out a cell proliferation assay, and these cells with very little ENaC divided very quickly, whereas when we put in extra ENaC, either the alpha subunit alone or all 3 subunits, we got a significant lowering in the cell division rate. So this gave us some confidence that ENaC might be playing a role in breast cancer, and we've just recently had our first paper accepted on this work, and we hope to carry on, and as Professor Hyland said, potentially looking at ENaC as a possible target for therapeutic intervention. All right, so that's it for your ENaC education. I have one more slide, and that's my acknowledgement slide, which, yes, it's big. Probably doesn't cover everyone who has helped me in this journey, but it's your students who drive a lot of this work, and there's a lot of students listed here. They've all contributed enormously to my own success, and I thank them, as well as many research staff who I've had in the lab, my colleagues and collaborators, and people along the way who've helped. I'd also like to thank my wonderful family. I was allowed to put this picture in. So this is my husband, Graham, and real particular thanks to him. He shouldered a lot of the childcare over many years. And our fabulous children, Daniel, Laura, and Matthew. This is us walking the Abel Tasman in January. Then I found a picture from the 1980s, I think. So a younger version of my parents, and my sister Claire and our sister Lauren, and my nana, who would have loved to be here today. And I'll also shout out to people online. I know Graham's parents are listening as well. Finally, thank you to all of you for being here, and thank you for listening to my story.
Kia ora, Professor McDonald. Thank you for a wonderful presentation. Kia ora koutou. My name is Pete Jones, and I'm the Deputy HOD Research for the Department of Physiology. So most of you will know that usually it falls to the HOD to give these formal thanks, but tonight that privilege falls to me. I think it was felt that it would probably be a little bit disingenuous if Fiona stood here and thanked herself for the excellent lecture, but I think most people would agree and not have complaints about that. A personal thanks as well, Fiona, because last night when I told my son, Rowan, that I was going to be late, he said, what was I going to do? So I said I was here and I was thanking someone for a good lecture. And so he said, well, what happens if it isn't a good lecture? Do you still have to say thank you? And I hadn't really crossed my mind, but well, probably. And then you got that look of, well, that makes you a liar. So thank you for not making me a liar tonight. It's also great coming to these because you get to hear the story of people as they progress through their careers, not just what they're working on now. And it was great to see Fiona as the small girl growing up in rural Otago. I hadn't realized, I hadn't caught on, but after hearing you say iron multiple times, I now could have recognized the rolling R, which I can't do. But then also how you progressed to where you are today. For those 400 level students out there, you might have noticed that Fiona had a publication from her honours degree. Those in physiology, that is now an expectation. But seriously, that's an extraordinary achievement, and it shows just where Fiona's research was, even at that early age. It was also really nice to hear when Fiona's passion for ion channels came about. And as another ion channel aficionado, it's great to see someone's excitement and passion for working on this research. The only comment would be, you've almost made ENAC sound as exciting as calcium channels. Almost. But beyond the science itself and those really fundamental discoveries that Fiona made in understanding ENAC, it was great to see her work in teaching, and that came across in the delivery of the lecture as well, covering really complex material in a way that a general audience can understand. I think that's also shown by the number of students that were on your last slide, how many you've mentored over the years, never mind the thousands of students which have come through physiology. And lastly, I just want to touch on the final kind of aspect of a professor, or a well-rounded professor, and that's the service to the university. Fiona's service as an HOD has been exceptional, and it's been really great working with her, under her. It's made life for a lot of us very easy, and I think Fiona's also had to work through some of the largest changes and challenges the department has probably experienced in the last several decades. Most notably with COVID-19 last year, and I don't think without her leadership the department would be quite where it is now. So all that leaves me to do is to ask you to join me in formally thanking Professor McDonald again, and to present her with a small token to remember the occasion. Congratulations, Fiona.
Kia ora, tēnā tātou koutou. My name's Paul Brunton and I'm the PVC for Health Sciences here at the University of Otago. I have two duties tonight. The first duty is to thank you, our audience, for coming to Fiona's lecture. These are really special events and they're made very special by the people who take the time to come and join us to celebrate the success of a newly appointed professor. Lastly, my final duty is to invite you to join us for some light refreshments over in the staff club. So thank you once again, and before you go, I think we should give Fiona another round of applause.